I'll just drag and drop these raw files, and you can drag and drop from any screen. So I'm not even on the Spectre screen right now, and I was able to drag and drop all of those. And then I actually don't have a database on this laptop, but that's fine. I don't actually need it to set this up. So these raw files are a little bit confusingly named. They're not really indicative of what is in the sample. But the way this is set up, this data set is set up, is you have a set of files. They each contain human and yeast lysate mixed together. Each sample has human and yeast mixed together. But the yeast amount is in different like amounts depending on the file. So there's three sort of quote unquote conditions. There's the 1x, which is sort of like the baseline, this like amount of yeast. And there's another set of files where it's double the amount of yeast lysate, and another set of files where it's triple the amount of yeast lysate. So you should see the amount of yeast intensities going up, but the human should stay constant if quantification is doing its job properly. And so the way, you actually don't have to set up what this experimental design to do a quantitative analysis, but it's better if you do, because it lets you do normalization, and I'll explain what that is in a second. But you can, I just memorized what these files are, but this is part of the 3x condition. So it's 3 files in the 3x. There's 4 files in the 2x, and then 4 in the 1x. And then what you can also do is if you already have this kind of set up, you could copy and paste from Excel. So I'll do that. So I'll just copy paste it into there, and that takes in comma delimited or tab delimited cells. So the way this is set up is for each condition, you have to have sequential, basically sample numbers, biological replicate numbers, starting with 1, and then they have to be sequential. And then, let's say you have fractionated data, right? If you have 1 biological rep that's fractionated, you would, let's just say these are fractions of this sample. You'd have fraction 1, 2, 3, 4, and you could have technical replicates too. So it's sort of almost a tree structure. I hope that makes sense. But this is not fractionated, so it's not as complicated. I'll copy and paste mine in. And if you do mess something up, let's say you accidentally put in like 4 or something, and you try to save it, it'll tell you biological rep 2 is missing in condition 3. So it was expecting a 2 here. The condition can be named anything. It's just a string. There's no format needed for that. It's whatever you want to call it. But these all need integer numbers. So save, it'll check obviously to see if everything's valid, and then there was no error message. So at least unless we mislabeled things in a acceptable order, then it's okay. So so I've got my experimental design set up. I'm also going to create my search task, which includes quantification. So match between runs will try to find peptides that weren't fragmented and identified in other samples. So what that means is, like, if you have one run where a peptide was fragmented and ID'd, and let's say in another sample it wasn't, the peptide is in the MS1 space as a chromatographic peak, but it wasn't fragmented. FlashLFQ will try to map this peak onto that peak and say this is the same peptide. That's what match between runs is. And that uses retention time alignment between samples. And then normalization is an intensity correction, basically, where there's typically some drift in measurements between samples. So like if you run 3 samples in a row, even the same sample, even if you're in the same sample 3 times in a row, the instrument can drift a little bit, and the intensities can decrease over time. And the other thing is that if you're running even seemingly identical samples, but they're different samples, there can be sometimes very slight systematic differences in how much like total lysate you inject or whatever. So normalization basically just finds if you're looking at two files that are in the same condition, or two biological replicates that are in the same condition anyway, it'll subtract peptide intensities between those two files, find the median difference, and set that to be 0. So the assumption there is that even between conditions, the assumption is that the median peptide is not changing in abundance between those samples. And that's a pretty common assumption when you're talking about normalization methods, but we use median centering. So I'm not going to run it because I forgot to download my databases, but I also have it already done. So I'm going to close this. Do you have any questions about setting that up?
No, I mean, this is, you're explaining it well. You're hitting things that I wouldn't have even asked, like whether or not it was median or mean centered. So yeah. I mean, I appreciate that.
Yeah, I tend to use median for a lot of things because it's just much more robust to weird tails and stuff. But I guess I should also say it when you save your experimental design, it will save it as a TSV file. I've already got it open. So you can look at the various files and then the condition and biorep, et cetera, et cetera. When you do calibration, it automatically updates those names with the calibrated MZML name for you. Okay, so I'll show you the quant output. So let's look at peptides first. That's pretty simple. Okay. So here we've got the peptide sequence that was quantified, and that tells you also what the base sequence was if this was modified. So for example, here's a carbametamethyl C modified peptide, but here just displays the base sequence along with this protein and gene name. And then you can see the intensities of the peptide across files. I don't know what is going on with Slack today.
Really popular.
And you can see so this one's probably a little messed up. You can see this is supposed to be a yeast peptide, so maybe this one's pretty noisy, but there should be a sort of a systematic change there, but the proteins are a little bit more obvious because there's more data per protein than there is per peptide. And this tells you if the peptide was quantified with MSMS, meaning it was fragmented in that file and the peak was found, or if it was quantified with match between runs, or if it just wasn't detected at all. There are also some errors where if multiple peptides find the same chromatographic peak, FlashLFQ will just cancel out that peak and says don't use this for quantification. Something's weird with that peak, and that happens a lot with phosphorylated peptides. Is if you have a peptide that's phosphorylated here versus here, they'll kind of collude. It's hard to distinguish those. If you want to look at the peaks, this might take a little while to open. It's kind of large. So this tells you the actual chromatographic peak information. Usually this isn't really necessary to look at. It's just kind of interesting if you're looking at if you're interested in the chromatography. It'll tell you where the peak starts. Peak starts the apex where the peak kind of crests, and then where the peak ends in the MS1 space. And then some other stuff like how that peak was detected, Also, I should say how FlashLFQ kind of works is, so MetaMorpheus will feed in the PSMs that were reliably identified into FlashLFQ. And then FlashLFQ will start at that MS2 fragmentation event and then look around that place for the MS1 peak. So this PSM's mapped column, what that means is like this peptide was fragmented, for example, twice, and both those PSMs found the same chromatographic peak. Whereas this one, let's say, was fragmented 11 times and one peak was found where all 11 of those mapped to the same peak. So there's no like double counting or whatever going on. There's some ppm error and other interesting measurements there. And there's also some ambiguity when, some PSMs find the same peak. So here this peak was ambiguous between this peptide and this peptide. And then probably what people usually care about is the protein groups file, which Rachel showed earlier. So here, this is a quantitative experiment, obviously. So we've got multiple intensities. And so these are all human at the top here, but if you scroll down a little bit, you'll find a yeast file or yeast protein. And if we plot like the intensities across the files, you'll see that there's three files that are sort of high, 4 that are sort of moderate, and 4 that are low, which matches what we'd expect from our experimental design. So the quantification, at least for this one protein, looks pretty good. And there's obviously some noise in between. So typically what you are looking for in a quantitative kind of LFQ context is what's called a fold change. So how much a protein changes between samples. That's because this is a relative quantification. It's really not an absolute quantification. So I won't really go into that much, but so if you, for example, take an intensity in the 3x condition and divide that by an intensity in the 1x condition, I think I might have gotten that wrong. That's not right. So this is human. This is a human protein. That's why it's not. That's why it's about 1. So it actually should be about that. You find a yeast protein. Just highlight this. And then like this, for example, divided by that, it's around 4. So again, around 3 block with some noise. So that can result in some pretty large numbers or very small numbers. So typically these are your log transformed, just to make them a little bit more easy to plot, really, is what it usually is. So if we take log transform that, log transform that, this is log base 10. I usually use log base 2, but it's a little bit more intuitive. So this minus this will give you a log fold change. In this case, it's log base 10. And because Excel can sometimes can mess up a little bit, you can give these error messages if there's like no measurements, because sometimes there are missing measurements, so missing values. I'm going to change this a little bit and say if there's an error. Input A. And that'll let us plot it in Excel properly. So we can make a little histogram. And you can see like there's a peak around 0. Those are the human proteins. And there's a peak around log base 10 of maybe 0.4 or so. And that's the yeast proteins that are changing. So another cool way to look at it is you can plot it. I've already pre-made some graphs. So I'll just show you like, here we go. So I don't know if this thing's in the way or not, but I actually don't know how to, oh, there we go. So the x-axis is the intensity of the protein, and the y-axis is the difference in intensities between the two conditions for various proteins. And so the black dots are human proteins, and the blue dots are yeast proteins. And it's around, this is log base 2 of like 1.58 or something like that. That's around that's a threefold change. And you can see at lower intensities, there's more noise. That's just a signal to noise thing. At higher signal, there's less relative noise, and at lower signal, there's more relative noise. So I think it's kind of a nice way to visualize this. Of course, in a real sample, like if you're analyzing cancer, for example, if you're doing like tumor versus no tumor, it'll look more like the black, and then there'll be a couple dots out here that might be differentially expressed proteins. You won't get a nice, separate distribution in real life when you're analyzing these real samples. But this is just kind of showing that the quant does actually work. That's why the spike and stuff is nice. You can kind of tell which proteins are incorrectly quantified and which ones are correctly quantified. And so we use Perseus a lot to do like t-tests and stuff. So you can actually import FlashLFQ results directly into Perseus. This is an older version, which I like more than the current one. But you can do a generic matrix upload from our TSV, our protein groups TSV. I won't go through all the t-testing and stuff, but so this isn't really a Perseus tutorial, but you can add the intensities to the main thing. I'm gonna get rid of this. You can add the organism to the categorical, and then the protein and gene to text. And the reason why I made organism and categorical in this case is because there's two categories here. One's yeast, one's human. Yeast changing, humans not. So if you want to label stuff like I did in the previous chart, it's a little bit easier to make that a categorical thing. So that's it. That's all you have to do to upload data into Perseus, and you can log transform and filter by valid values, because there are some down here that have quite a few missing values, and then you can impute and then do t-tests and stuff. And I can go through that if you want. It takes a couple minutes, but it's not too bad. And then with this data set, I think we, at an FDR of like 5%, I think there's actually like maybe on 8% or so that are significantly changing, that are human, so they're incorrectly changing. But the FDR control is pretty good for like t-tests and stuff. It's not amazing, but it's not bad. So that's kind of like the basics of quant.